Okay, so welcome to this next video in which we're discussing antisense oligonucleotides. Okay, so these are going to be a way of stopping the expression of a gene. Okay, so let's now talk about what an antisense oligonucleotide is. So, we have looked at the central dogma. Okay, we have seen that the coding strand of a gene is going to be transcribed into a sense piece of mRNA, which will then go into the cytoplasm and be translated. Okay, so say we want to stop the expression of this gene, then what we could do is try and take out this piece of mRNA before it can be translated. Okay, and the way that you can do that is create little structures which are complementary to sequences of organic bases within the sense mRNA strand. So here is our sense mRNA strand here. Okay, now we can create little strands, okay, little small strands which have a complementary sequence of organic bases to the organic bases in the mRNA. Okay, now the key thing is that these little single strands here that are complementary to the sequence of organic bases on this sense mRNA, it's going to be a single strand of DNA. Okay, so let me repeat that. This little strand that we're going to use to bind to a portion of the sense mRNA is going to be DNA. It's single-stranded DNA. Antisense oligonucleotides are small fragments of DNA which are single-stranded and have a complementary sequence of organic bases to a certain sense mRNA, okay, or a certain portion of a sense mRNA. Now these are small little things, okay, so they're usually between 13 and 25 organic bases. So the whole sense mRNA strand will be far bigger than 13 to 25 organic bases. Okay, so this is only going to bind to a region of it. Okay, now it is an oligonucleotide because oligo means some or a few. So it's a few nucleotides polymerized together here. And basically, it is antisense because the sequence of organic bases that it has will be complementary to these this small sequence of organic bases on the sense mRNA, and therefore actually it will have the same sequence of organic bases as a portion of this antisense strand down here. Okay, therefore it's an antisense oligonucleotide. Now, what's the significance of this? Well, these antisense oligonucleotides, we can produce little single strands of DNA which are complementary to certain target mRNAs, and then if you expose a cell to these, what will happen is it will go inside the cell, it will bind to these mRNAs, and what it's going to do is stop that mRNA from actually being translated. Now, how does it stop the mRNA from being translated? Well, there are two major divisions for how it can do this. The first is that it can work via an enzyme known as Rnase, okay, H. Okay, so Rnase H. So, what is Rnase H? Well, let me go over the page and tell you about Rnase H. So, Rnase H is an enzyme which degrades RNA-DNA hybrids, okay? So, when you have a piece of mRNA, so let's get our sense mRNA back here again. So this is our sense strand of mRNA, okay? And here is our little now antisense oligonucleotide, which is DNA, remember, which is bound to it, okay? So you have now got what is known as an RNA-DNA hybrid because you've got RNA bound to DNA. Okay, now the enzyme Rnase H, which I'm showing here, recognizes RNA-DNA hybrids and breaks down the RNA strand that is attached to the DNA. Okay, so let me show this happening. So we'll colour in Rnase H in purple here. And basically it is what is known as an endonuclease. So let me explain what an endonuclease is. An endonuclease is something which breaks bonds between nucleotides, and it breaks them from deep within a strand. So let me explain this. So basically, let me firstly just explain what a nuclease is. A nuclease is anything which is going to break this bond between the third carbon atom's alcohol group and the phosphate group. So nucleases break these bonds here between nucleotides, basically. Now, let me explain what the difference between an exonuclease and an endonuclease is. So, if you have a great big long strand of mRNA here, an exonuclease would start right at the tip down here and break off the very end nucleotide, basically. So this is what an exonuclease would do. It would work from the exteriors, basically, rather than the interiors. Okay, whereas an endonuclease would go right to the middle, or anywhere in between, and just break the bonds between the two nucleotides in the middle there. Okay, this is what an endonuclease would do. So exonucleases remove nucleotides from the ends of the nucleic acids, whereas the endonucleases, they chop nucleic acids up from the middle, basically. Okay, so RNase H is an endonuclease. So when it finds this RNA-DNA hybrid here, what it's going to do is start chopping up the RNA that's bound to that DNA fragment here. So it won't chop up these bits that don't have the DNA bound, but it will chop up these pieces where the DNA antisense oligonucleotide is bound. So it might chop here, etc. basically. So what you'll end up with is multiple fragments. You'll end up with the mRNA being chopped up into pieces and destroyed. Okay, it's not going to be able to be translated now because it's been broken up by this Rnase H enzyme. Now, this enzyme is actually very, very important with regards to the pathology of the human immunodeficiency virus. Human immunodeficiency virus is what is known as a retrovirus. Okay, so it has a genome that is made up of mRNA. Okay, so let's say this is a piece of mRNA here. Okay, so here is the genome of HIV, and then what HIV does is it synthesizes a piece of complementary DNA to the RNA genome once it actually gets into a human cell. So it uses the enzyme reverse transcriptase, which synthesizes a complementary piece of DNA known as a cDNA strand. Okay, so I'll label it up as a cDNA strand. So this is a cDNA strand. Okay, and then what you have within the cell is a cDNA strand bound to the original mRNA or just RNA template that was inside the HIV virion. Okay, now what will happen is the Rnase H enzyme will break down this original piece of mRNA which is bound to the cDNA. That will disintegrate apart basically, and then you can move on to synthesize a complementary strand to this piece of DNA that you've synthesized. Okay, so you'll make another piece of DNA, and now you've got a double-stranded piece of DNA which can then be implanted into the human genome, and that's very important in the pathology of HIV. Okay, so this is happening in HIV, for instance, human immunodeficiency virus. So, one way that these DNA antisense oligonucleotides work is that when they form this DNA-RNA hybrid, that is then the target for this Rnase H enzyme, which is an endonuclease, and will basically break up the mRNA into loads of pieces which then aren't going to be able to be translated, and therefore you've done away with it, basically. Okay, however, there are also other mechanisms which are known as collectively RNase H independent mechanisms. So RNase H independent mechanisms, and these are far less well understood. Okay, so we don't really understand how the antisense oligonucleotides work if they don't work with RNase H, and we will see examples of antisense oligonucleotides which don't actually activate RNase H, i.e. RNase H will not recognize the double strand where you've got the mRNA bound to the oligonucleotide. It won't recognize this as a DNA-RNA hybrid in some cases, and therefore you don't get the activation of RNase H. So some of them only work through RNase H independent mechanisms, okay, and these aren't nearly as well understood. One of the main ideas, of course, is that if you've got this antisense oligonucleotide bound, surely that will stop the translation of the mRNA, because simply as the mRNA is going through the ribosome, this will get caught, because only single-stranded nucleic acids can go through a ribosome, and it just won't budge basically, and the ribosome will therefore get stuck and just can't proceed because it can't bring in this portion where you've got a double-stranded nucleic acid, and that will stop the process of translation. Okay, so that's one of the main ideas for how RNase H independent mechanisms work. The point is that these antisense oligonucleotides are going to kill the expression of this gene. Okay, now in the next video what we'll turn our attention to is the different types of antisense oligonucleotides that you can use and their various pros and cons.